We'll move now to the next presentation by Mr. Faraci on, Mark Faraci, not F, on hypertension and aging. Mark is from the University of Iowa, and he's going to show us some very interesting data with, in particular, the ACE polymorphism.
And this works. What do I need to do to work this? Is there a secret? The middle? Oh, there it is, okay. Okay. So I don't need to, well, first I want to thank the organizers very much for having me come and present at this meeting, and it's been a great day so far, and I've already learned quite a lot, so I'm really happy to be here. I don't need to say anything more about how important hypertension after the previous talk was, so I can save time on that. What I want to do in the time that I have is to talk a little bit about some of the basic studies that we've done and a few others related to the impact of hypertension on cerebrovascular disease and some of the underlying mechanisms. One of the take-home messages I hope that you get is that that all hypertension is not created equal, and that the impact on the cerebral vasculature depends on the underlying mechanisms that are involved in the hypertension. I'm going to give a couple of examples of sort of some mechanisms that interact with or may be common between aging and hypertension under conditions with aging where there's no elevation in pressure. There are some of the same mechanisms that may be involved. And then the third thing will be some information related to a system that we think may play an important role in vascular protection, and that's the PPAR gamma system. And I'll explain as we go along why we got interested in that and what we've learned about it recently in specific vascular cells. So this first slide, I've broken down kind of somewhat arbitrarily, but I hope it makes a bit of sense. Some of the key features of what I call large and small vessel disease. As you'll see, we've heard about a number of these already today. Some of them, like endothelial dysfunction, has been mentioned, and that's common in both large and small vessel disease. Other things, although this is somewhat arbitrary, it's not an absolute breakdown. Things like atherosclerosis tend to be much more prominent in large vessels. Things like inward vascular remodeling, which I'll show an example of, tend to be more important in very small vessels. So several of these key things I want to give some examples of as we go along. We know that most of the time what we're concerned about and interested in is the problems that involve reductions in perfusion, sometimes of moderate magnitude and sometimes of quite severe. There are a few clinical conditions where you get hyperperfusion, but most of the time we're worried about flow reductions and what accounts for that. And we, of course, know what the implications are. As part of our studies of endothelial dysfunction, and a lot of what I'll talk about today focuses on that cell type. For those of you who don't study endothelial cells, it has an enormous impact on the vascular wall, and also we know now onto a number of non-vascular cells within the brain. Some people have made the comment that the health of the endothelial cell sort of determines the health of the rest of the vasculature. So it's a key sort of cornerstone cell in that sense. And we have clinical data that abnormalities in endothelial function, and even specific ones that are involved in regulation of vasomotor tone, and I'll show some examples of that, is predictive of clinical events, including stroke. So what happens in the endothelial cell has very broad consequences, even though sometimes experimentally, to try to define mechanism, we may only look at one aspect of it, like how it regulates vasomotor tone. It's involved in many things. The system that we focused on largely related to hypertension is the angiotensin system. And I'm sure everybody's familiar with the basics of this system. This peptide that's produced here in the center is sort of the key one that you're interested in that mediates most of the vasoactive effects. Precursor peptides are thought to be biologically inactive. And at least for the detrimental effects of the renin-angiotensin system, they're thought to very predominantly be through the AT1 receptor. And they're varied. There's certainly major effects on vascular tone, and that's one of the reasons why they think angiotensin contributes to blood pressure increases. But angiotensin, independent of its effects on pressure, is pro-inflammatory, it's pro-oxidant, has effects on vascular permeability, and it has effects on growth of the vessel wall and vascular remodeling. I'll show some examples of that. So this is one of the, at least for using mice, one of the earliest studies that we did to look at the, a model of angiotensin-dependent hypertension in a mouse and what effects does it have on the vasculature. And in this case, we've used acetylcholine, which we've heard mention of today, a nitric oxide donor, and you look at the change in diameter of the vessels in these cerebral arteries, and it's greatly reduced to the animal that has angiotensin-dependent hypertension, the R plus A plus mouse. And if you use a scavenger of superoxide, pegylated superoxide dismutase, you can restore that response acutely to normal. So this is very early evidence that whatever is accounting for this endothelial dysfunction in this model of hypertension, it looks like oxidative stress is part of it. And we've spent quite a bit of time since then trying to tease apart mechanisms like what's causing the oxidative stress, other endogenous mechanisms to protect against it, and so on. So that's a genetic model. We've also done, and it's one of the most common models out there for people that use mice in hypertension research, is an equivalent thing, which is to infuse angiotensin systemically using osmotic mini pumps for a month. And you can mimic the kind of changes you get with lifelong hypertension in the genetic models. You can get marked changes in endothelial dysfunction and so on. Now, we've been interested mostly in both vascular tone regulation and vascular remodeling, the things I mentioned earlier. I've already hinted about how there's this interrelationship between pro-inflammatory and pro-oxidant mechanisms. And these, what I've highlighted on this slide, are some of the mechanisms that we've studied. This isn't meant to be sort of complete for the entire field of angiotensin research, but there's some of the things that we've looked at in the cerebral vasculature and that seem to be important in determining the phenotype. There's some key sources of superoxide, such as NADPH oxidase, which I'll show an example of that a couple of times. Some key specific antioxidants that we've identified that are normally protecting against these processes, and it may be failure of this protection that contributes also to the pathology of the disease. And then there's, on the inflammatory side, or inflammatory-related mechanisms, things like IL-6 production is critical for some of these vascular abnormalities, and things like the signaling molecule STAT3. So I'll show a couple of examples, one sort of on each side as we go through. So here's another experiment where we made a mouse chronically hypertensive with angiotensin, and you get the reduction in endothelial function. And if we treat the animal with a small molecule inhibitor of STAT3, the transcription factor, which is thought to work primarily on the pro-inflammatory side, but also may crosstalk with some of the pro-oxidant mechanisms, you can largely prevent that effect on the vascular endothelial function. If you measure superoxide, measure one of the free radicals that's being produced, it goes up with angiotensin, and that's also prevented. So it's just one example of part of the mechanism that is involved in this process. Now, one of the things that we've been interested in as far as the impact of hypertension for a number of years, and these are studies that we typically do in collaboration with Gary Bombach, is the effect of hypertension on inward remodeling. And this is something that's fairly, it's not an absolute thing that it only happens in hypertension, but it's one of the key features of the microcirculation or small vessels during hypertension. And it's where vessels in the brain, for example, the small P arterioles on the surface, and now there's evidence that within the parenchyma as well, there's a three-dimensional rearrangement of the vessel wall so that the lumen becomes smaller. Even if the vessels are maximally dilated, you have a permanent inward remodeling. And you can see that experimentally in some of the some rat models, in that first mouse model that I mentioned, the chronic genetic model of angiotensin hypertension. If you infuse angiotensin chronically for a few weeks, it'll produce the effect. And in patients, when they've looked at the brains from patients with essential hypertension, although the way the data is quantified is perhaps not quite as precise as it should be, there's certainly evidence that the same kind of process is going on within the parenchymal arterioles in the human brain with hypertension. The physiological consequences you can easily imagine. A vessel that even when maximally dilated is smaller is going to have a major impact on regulation of microvascular pressure, so the local perfusion pressure that these vessels are seeing, like as they dive into the parenchyma, it's going to affect vasodilator reserve, going to raise minimal resistance, and obviously it's going to affect flow potentially normally, especially with things like ischemic events when you're trying to get dependent on collateral flow and so on, and your collaterals are smaller, can have a major impact. So I want to show you some examples of what can happen in hypertension. So here's an experiment, whoops, keep hitting the wrong button, I'm sorry. Two different hypertensive mice in this slide. The one that I've already talked about, this R plus A plus mouse, which is angiotensin II dependent, and then a mouse called the BPH2 mouse strain. It's angiotensin II independent. If you look at their blood pressure in conscious animals, they have equal hypertension. So no difference there. If you look at the very small arterioles in the brain and look at the cross-sectional area, the thickness of the vessel wall, they have equal increase in thickness. So their hypertrophy, if you will, is the same. But look at what happens with the diameter in maximally dilated vessels. If it's angiotensin dependent, the vessels get much smaller. And if it's angiotensin independent, there's no change. So it would suggest that angiotensin dependent hypertension is going to be a lot worse for the brain, or make this animal be predisposed to a worse outcome. If you had an ischemic event, if you're trying to keep normal neurovascular coupling going year after year and so on. So all hypertension is not the same as far as the vasculature. What kind of mechanisms underlie these kind of changes? One of the things that vascular people have focused on to a great extent in recent years is this enzyme is a source of superoxide ion, the NADPH oxidase, if you're not familiar with it. Thought to be a major source of free radicals in vascular cells. There's multiple isoforms of it, but at least so far the data that we have is that the NOX2 isoform is the predominant one as far as accounting for most of the phenotype. So these are experiments that were done in wild type and NOX2 deficient animals that were either infused with vehicle or infused with angiotensin for a few weeks. You get an equal rise in blood pressure, whether NOX2 is present or not. You get endothelial dysfunction in the wild type, if you look at acetylcholine, and that's absent in the knockout animal. So even though he's hypertensive, his endothelial function looks good. Nitroprusside is normal. In the wild type, you can see increase in expression of components of this oxidase. You can measure increases in superoxide. And you see that inward remodeling in this animal. The vessels get smaller, and in the NOX2 deficient animal, you don't see that reduction. So not only does it seem to have an impact on regulation of vasomotor tone, that same enzyme seems to be critical for some of the vascular structural changes. And this NOX2 is not, it may be a broad player in cerebral vascular disease. It goes beyond hypertension. So this is some recent data that we did in a different model, animals that have diet-induced obesity. So you put mice on a high-fat diet, they gain weight, whether you look at total body weight or you look at the deposition of fat. Those changes in weight gain and obesity are not affected by the deletion of that NOX2 isoform. If you look at the small arterioles, over time, it takes about 12 weeks, you get pretty good endothelial dysfunction. If you do that same experiment down here, no endothelial dysfunction. So they become just as obese, and some of their other metabolic changes are the same, but you protect the vasculature when you target this oxidase and remove it genetically. And the other interesting thing from this data, and it wasn't something we really expected, is you're seeing some fairly prominent microvascular effects relatively early. If you look at the same time in carotid arteries from those same animals and look at their endothelial dysfunction, nothing's happened yet. So the dysfunction that you see is occurring much earlier in microvessels in the brain. You have to go much, much longer to begin to see dysfunction outside of the brain. So in a way, the microcirculation is sort of predisposed to this kind of dysfunction. Now, we think that there's some links between vascular disease of different origins and some common underlying mechanisms between aging and hypertension. And I want to show you one example from a study we published a couple of years ago. So we looked at cerebral arteries, and on the left side, the left side here is just wild-type animals at 3 different ages: adults, animals about 1.5 years, and then animals that are almost 2 years old. If you look in the aorta, you don't see very much. In fact, the aorta, some of the animals are even older. You can go almost to 2 years before you start to see much of a change in endothelial dysfunction. And then if they get even older than that, it becomes more progressive. But at only 1.5 years in cerebral arteries, you've got a pretty profound effect. So the responses are reduced more than half at a time when the aorta looks pretty normal. And if you were to look at carotid arteries at the same time, they would also look normal. So it's another example where the cerebral vascular disease seems to be occurring more rapidly in an accelerated rate, if you will, than what's seen in vessels outside of the brain. Now, could angiotensin be involved? These animals are normotensive, but we had reason to believe that it still might be involved. So we repeated the experiments in animals that lack the AT1 receptor. So this is an additional wild type group, roughly the same age for the adults, a little over a year and a half. And where here you got this nice impairment, more than half. You see that in this strain as well, and they're completely protected. So in the absence of hypertension, this renin-angiotensin system and that receptor is still having a major effect, at least on this endpoint, this endothelial dysfunction in the cerebral vasculature. So as these studies went along, we also were sort of always thinking about mechanisms that might be there endogenously to buffer or protect against some of these kind of changes. And if we have better insight into those, we may be able to manipulate the system and take advantage of that, sort of the equivalent of neuroprotection type studies, but focusing on the vasculature. And we got interested in the transcription factor PPAR gamma for these experiments. It's the target of the TZD drugs used to treat diabetes, and it's known through its effects on its target genes to have a number of diverse effects within the body. Some of them are metabolic, some of them are renal, some of them are vascular. You can improve vascular function in some models. You can lower blood pressure in patients that have hypertension and so on. But we specifically were interested in, what is potentially its role in vascular cells. And at the time we started this, there wasn't so much known. Some of the earliest data that we got, which suggested that this might be worth pursuing, is that we looked in this chronically hypertensive mouse model, which has some impairment of endothelial dysfunction. We've already seen examples of that. If we treated with pharmacologically, treat the whole animal with one of these agents that activates PPAR gamma, we saw a beneficial effect. So that at least encouraged us to go on and to do some more sophisticated genetic studies to begin to look at the impact of this transcription factor within vascular cells. So how did we do that? Well, there's a paper that came out in Nature a little while back that described some dominant negative mutations in PPAR gamma in the ligand binding domain of the molecule. And these patients were insulin resistant, diabetic, had early onset hypertension and so on. And they also had some other metabolic abnormalities. And the thing that attracted us to this paper was wondering whether, could the hypertension be due to a vascular problem? Some of the other metabolic problems could be due to something else, but could the hypertension be due to vascular? And was PPAR important within vascular cells? So to address this, my collaborator, Kurt Sigmund, one of the students in his lab, made an animal that has, took that human gene with one of the dominant negative mutations and put it under control of a smooth muscle specific promoter, and then made mice that we could study. Mice that these SP467L mice, which are expressing the human dominant negative mutation. Unlike patients that have these mutations, they have normal glucose, normal insulin. The metabolism is normal because you've done a vascular specific manipulation. And the thing that we kind of speculated we might see, the hypertension, was in fact there. They're modestly hypertensive. So we took vessels from these animals and to begin to look at the role of PPAR gamma specifically in vascular smooth muscle. And this is the kind of data that we got initially. These are studies done by Michael Da Silva in my lab, a postdoc. Non-transgenic controls, the animal with the dominant negative mutation, and then we also had animals that overexpress wild-type PPAR gamma, have an increased level. And their baseline characteristics are very similar, but one of the key features that we saw early on is that they have a greatly impaired response to NO, that protective molecule that the endothelial cell makes that does so many good things for the vessel wall. Greatly impaired in the animal with the dominant negative mutation. It doesn't matter if you use an NO donor like nitroprusside, or if you use agents like acetylcholine or angiotensin 1,7, which cause release of endogenously produced NO, greatly impaired response. And if you overexpress wild type, it didn't seem to do anything, if you look over here. If we look in the very small microvessels in vivo, this is not just an in vitro sort of artifact. This is in vivo data using those same approaches, looking to acetylcholine nitroprusside, greatly impaired responses. So it's also occurring in the very small microvessels. So, I mentioned that the PPAR gamma mouse has some moderate hypertension. Blood pressure is increased by about 10 millimeters of mercury. If we increase blood pressure by that amount in a mouse using another approach, using DOCA salt, get the same level of hypertension, the vascular phenotype is very different from what you see in the PPAR mouse. If they've got lots of endothelial dysfunction, so the acetylcholine responses are greatly impaired, whether it's in small arterioles or in the middle cerebral artery, but the responses to NO itself are normal. So the mild hypertension cannot explain this great reduction in the response to NO and NO signaling. So what mechanisms may explain it? And there's two that we sort of went after initially that we thought might be responsible. One relates to oxidative stress, and I've already given some examples of that, whether things like superoxide made by NADPH oxidase, which is known to activate Rho kinase, might be involved. But Rho kinase could also be involved in other ways. So it was sort of an attractive candidate for a way that vascular tone could be abnormal in a model like this. And there were some hints in the literature that PPAR gamma, there may be some links between that with Rho kinase. So we did some experiments to look at that. The top two panels we used are basically looking for a role for oxidative stress. We used a superoxide scavenger. We used a PARP inhibitor, which is a downstream target of oxidative stress. No improvement at all in these responses. If we used an inhibitor of Rho kinase, however, this greatly impaired response to an NO donor or to endogenous NO is restored essentially to normal. So Rho kinase seems to be key, suggesting that perhaps normally PPAR gamma is suppressing the activity of Rho kinase. When you interfere with it genetically with this dominant negative mutation, its activity becomes much higher, and then you get these abnormalities. It extends beyond just endothelial dysfunction. So you look at myogenic tone. It's also elevated in these animals shown here on the bottom left slide. And then we looked at the structure of the vessels. I mentioned the end-root remodeling earlier. At least at the level of the middle cerebral artery and its sort of first branch, not much of a change if you do a passive pressure diameter curve. I just noticed I forgot to put pressure on the bottom. I apologize for that. Not much of a shift in the curve. But if you look farther downstream, a couple of branches down to small microvessels, you get the same end-root remodeling. So here's the non-transgenic over here, and then the transgenic animal here. So over a range of microvascular pressures, these very small vessels, and this is when they're maximally dilated, are much smaller. So in addition to hypertension that can cause end-root remodeling, this is actually the first example that we found of something that could mimic hypertension and sort of without really having a hypertension component. So genetic interference with PPAR gamma has similar effects on vascular structure as activation of the renin-angiotensin system. We wanted to look just a little bit more about where other elements of the renin-angiotensin system activated. For that, let me go back for 1 second. In addition to looking at the direct effects of angiotensin II, we also looked at the precursor peptide, wondering if possibly the metabolism or the breakdown, or not breakdown, but metabolism of angiotensin I could be affected. And really the key slides are on these top two panels here. There's a bit of a shift in the vasoconstrictor responses to angiotensin in the transgenic mouse, but a pretty big shift in the response to angiotensin I. And angiotensin I is normally thought to not really be biologically active. So we pursued that further in the next slide, trying to see, well, what could explain this 3 or even 4-fold increase in the response to angiotensin I. So here, we're giving angiotensin I now, the precursor peptide, not angiotensin II. You get a small response in the controls, you get a pretty big response in the transgenic, but you can completely block that with a drug that blocks the AT1 receptor. So it implies that the peptide had to be converted to angiotensin II, which then activated the receptor. And we could confirm that to at least to a large extent in this slide where we use captopril, which is an ACE inhibitor. And although it doesn't completely wipe out the response, suggesting there may be some other pathways involved in converting Ang1 to Ang2, you block a lot of the response. So it suggests that not only is the direct effects of angiotensin enhanced when you've interfered with PPAR gamma, but that the processing of the peptides may be enhanced. So this is a second way in which this agent may normally protect against angiotensin-dependent vascular changes. And then just with a couple more slides, if you'll give me the latitude, I want to talk about a couple of some very recent data we have in carotid vessels. And I want to show this because I think it's some interesting data conceptually and is going to lead to a future experiment that may have relevance to what I just talked about. We also have an animal that has PPAR gamma manipulated just in the endothelial cell. So that's sort of the mirror image. And in this animal, sort of everything's normal. Body weight, metabolic indices, blood pressure is normal. If you let these animals get old, first, in the adult animals, we saw a big effect of big baseline effect if you interfered with PPAR gamma and vascular muscle. The NO responses were way down, and so without any other intervention. Here, the baseline effects of like the transgenic and the non-transgenic, if you look at these acetylcholine curves, it's pretty similar. So it doesn't look like much has happened, just like the blood pressure didn't change and so on. But we thought that these animals might be predisposed if we either challenged them with angiotensin or in this case, we aged them. And that's in fact what we saw. Our old animals, shown here, the non-transgenics and so on, are a little less than 2 years old. And at that age, you don't see very much in the carotid arteries. You see a lot of impairment in this animal. So if you've interfered with PPAR gamma only in the endothelial cell, you get a lot of age-induced vascular dysfunction. Responses to the NO donor are normal. As far as the mechanism, I'll just skip this. I can just explain it, I think. One of the things that's been implicated is the cyclooxygenase system in aging abnormalities. We gave indomethacin, and you see it did nothing in these old transgenic animals, so no improvement whatsoever. If we give a scavenger of superoxides, we're back with the free radicals, we saw a big effect. So this is another difference. In the smooth muscle mouse, we gave scavengers of superoxide, and it was a negative result. We went on to rokinase. Here, it looks like oxidative stress is perhaps playing a big role. So by manipulating this cell, the underlying mechanisms, it depends on which cell you've altered. To look at this a little bit further, we're back to NADPH oxidase again. We use an inhibitor of these oxidases, and just like the superoxide scavenger, you can improve the response pretty much to normal just with acute treatment. And rokinase can be activated by reactive oxygen species. I mentioned that in passing earlier. So we wondered, because we had this other data, that within smooth muscle that rokinase might be important. And rokinase has interactions with the source of NO and endothelium. We used a rokinase inhibitor, and it also improved responses substantially. So another example of these interactions between PPAR gamma and rokinase, except in a different vascular cell and now involving oxidative stress. And so this is, I think, my last data slide. I showed you this experiment earlier where you get this sort of what I call accelerated vascular aging in the brain compared at least to the peripheral vessels. So one of the experiments that we want to do now is we have animals that have increased expression of wild-type PPAR gamma in the endothelial cell. And I think it could be very interesting to allow these animals to get older or to make them hypertensive, and then to look at not only at their vascular consequences, but then look within the parenchyma at changes that may occur and see if we do a very vascular specific manipulation, whether we can have a favorable outcome on other indices besides related to flow regulation. So I hope I've convinced you that PPAR gamma seems like a pretty interesting molecule for cerebral vasculature. 10 years ago at meetings, nobody at the sessions I went to at least was talking about PPAR gamma. And now we know that in the absence of these clinical drugs, that these dominant negative approaches suggest that this signaling pathway is active and it exerts some particularly prominent effects, sometimes cell-specific. In vascular muscle, it's affecting NO signaling, myogenic tone, and it protects against inward remodeling. In endothelium, it looks like it's essential to protect against a key part of vascular aging, the endothelial dysfunction that occurs. Genetic interference with PPAR gamma can mimic some of the effects of angiotensin, which is interesting and sort of gives us ideas about what pathways are maybe involved. So I think that either PPAR gamma itself, or as we're also trying to learn more about, what are its specific targets, may be some good things to look at in the future in relation to mechanisms to protect against either large or small vessel disease. And this is the last slide. Just, I think I've mentioned most of the people, Kurt for the help with the PPAR studies, and Gary for the inward remodeling, Michael for most of the PPAR work, and then our funding is on the right. And of course, I want to acknowledge the Duke Foundation. I've done a lot of collaborative work over the years at Iowa, and now that we have this new network to focus on small vessel disease, I'm beginning to work with a new group of investigators, and we're very excited to continue this and to try to get some additional insight into what kind of mechanisms underlie these changes in the smaller microvessels, whatever term you like. Thank you.